Hello and welcome to my new video, and it will be dedicated to a very important subject in molecular biology and microbiology and taxonomy as well. I will try to make type materials for herbarium deposition. Simply, when you describe a new taxa, new species or genus or family, you need to deposit culture material to the culture collections, and you need to put the herbarium specimen to the publicly available collections for other researchers which gonna want to check your type materials. And it's I think it came from zoological nomenclature to botany as well. And anyways, we need to do that. For that, we will need two parts for one envelope. One envelope like this. I will fold it later. Will have two parts. First part is gonna be glass vials. You can see them here, and actually it's very practical and important. You can see the barcode and number in the catalog. So since I'm gonna make two type materials, I need two vials, vials for two different type materials. And these vials I will use for preserved material, preserved biomass. And in algae it's better to use formaldehyde solution like this, and it should be labeled. It's nasty and it's 4% formaldehyde. I'm gonna put some biomass in the vials and put formaldehyde here. So this is the first part. Another part of type material is gonna be glass fiber filters. These ones, and you can see the catalog number, and some of them are here, and each of them we will use for 3 filters. We will use for one type material. It would be holotype for Maxicurus and epitype for Pluricapsa. And afterwards, when we're gonna put liquid biomass or biomass with liquid parts to these filters, we're gonna dry them on the petri dishes here. You can see petri dishes here. We're gonna put these filters into this envelope, and afterwards we're gonna put those ones in a huge envelope. And when we will send the envelope to the herbarium, they will print the labels with the notification that it is in effect holotype material. So we have everything for that. Let's move forward. I'm gonna put my phone here. I have two petri dishes with Pluricapsa minor, and I have accession number there, and I will put the glass fiber inside the petri dish, and I need to label petri dish. I usually label petri dish on the bottom on the petri dish, because you can lose the lid, and if you will put the glass fibers here, you will not lose them and you will not lose your label. So we're gonna label it. It's gonna be GSECHRMK 1707. I'm gonna note that it's Pluricapsa minor. Okay. And for that I'm gonna need forceps to grab glass fiber filters. And as I told before, I'm gonna need 3 glass fiber filters for each type material. So I got them here. And I will open the petri dish. I also need this wooden sticks to scrap the biomass from the surface of agar. And I just literally will put it on the top and press it a little bit. And I will use 1 petri dish with biomass for dried material, and another part will go to the formaldehyde solution, which will go to the glass vial. It's a long process and you should be very careful and not supposed to be in a rush, and you need to dedicate a fair amount of time for doing that. Because down the road, when some other researcher would like to extract DNA from your samples or just explore them under the microscope again, we're working with cyanobacteria, which are microscopic. You need to make cover slide for that. I think it's good enough. And why I'm having the petri dish with biomass, because I wanted to grow it, make clones of that. So right now I have in this petri dish with biomass, and it's pretty much done. So this part is done, and I will put the lid and I will put it in some dry place to dry it out. So we're gonna put it here. So right now I will make another part of type material, which is fixed formaldehyde vial. For that I'm gonna label it as well. It's GSC, GSE, SHR 17 sample cups. Okay, how I do that, I will put a little bit of formaldehyde, and actually it's probably better idea to work under the hood with formaldehyde. So I put a fair amount of formaldehyde to the vial, and I'm gonna make the same sort of things. I'm going to remove parafilm and see. So biomass here, which is good. Mostly formaldehyde material, biomass, usually used for DNA extraction, because after fixation in this nasty chemical, color of algae gonna be changed, and in terms of cyanobacteria, it's very important morphological feature. Color is important, and the color of cells is gonna change after inoculation in the formaldehyde. I don't have a lot of biomass, to be honest, for formaldehyde, but it's still better than nothing. Actually, since Pluricapsa is Coccoid taxa and it's not growing so fast, it's gonna be okay. So this part of procedure is done, and I close it. And actually, after I'm gonna parafilm the vial, because formaldehyde can be evaporated, and we don't need that. So, but here you can see some biomass moving. So I'm done with that, and another part of my video will be dealing with making type material of Myxocoris, and I'm gonna have culture on this land, and also culture on the liquid medium. And for taking this biomass from the liquid tube, I'm gonna use the vacuum machine. Also have it here. Okay, this is another part. And I'm gonna make the same amount of fiberglass filters. It's gonna be 3. One, 2, 3. By the way, it's not Fisher brand advertising. It's happened to be. So what I'm gonna use, I will use solid biomass for formaldehyde part, and I'm gonna use this one for filters. And for that, as I said, I'm gonna use vacuum machine. So we have thing with the vacuum here, and this bottle, we're gonna put this lid on the top, and I'm gonna put the glass fiber on the top of this machine. I'm gonna start to run vacuum, and you can feel it and hear it. And I basically will shake this tube a lot. Open this tube, and I will put a little bit of liquid into the filter. And you can see that all liquid things going down, and you have biomass on the filter. It's pretty much the basic idea of using vacuum pump for doing that. You can see the biomass on the top of the filter, and afterwards you just using the forceps, and you removing this filter and put it in the petri dish. That's pretty much it. I didn't have a lot of biomass for Myxocoris in the liquid tube, but I still have leftovers in the tube with liquid medium, and I still gonna use it. And I just will put it on the top of another fiberglass filter. It's actually pretty fun, and when you have a couple of samples, but if it's like 10 or more, it's not so exciting. I'm sorry guys that this video is taking so long, and but it's science and it should be precise. And for that I just will scrape some biomass from the top of the agar and then put to the filter. And to be honest, I didn't work with type materials of cyanobacteria from back to the days, but it's very problematic because a lot of museums, they have very biased and very old and not obvious type materials. That's why we want to make ours pretty good, and that's why we're using a lot of biomass, and we culture them for that reason. So I culture them for that particular reason. So I transfer them from culture to another tube to gain biomass for that. I will actually press a little bit of this biomass because I scrape a little bit of agar. I don't want to agar to be there, or I don't want the biomass to be detached from the glass fiber filters. And I'm gonna just press it down. It's pretty green. I like that. So we're pretty much done, except that I forgot to label this petri dish. I'm gonna do that, and once again, after the material is dried, we're gonna fold this envelope and fold it like this in very nice shape, and we're gonna put one glass vial with formaldehyde preserved material, and also with 3 fiberglass filters, which will go to this envelope. And all this material gonna go here, and we're gonna ship it to some herbarium, or we will deposit this thing in our own herbarium, and it's gonna have accession number for future researchers to have access to this exciting and very important material. Thank you for watching that, and I hope that it was very helpful. Thank you and good luck.